Good day to everyone. I'm Wong Yeng from UC Malaysia, Sarawak. I'm a plant systematist with a broad range of research interests involving the flora of the island of Borneo. These are from the forest. This is a selection of Calamus palm purchased on a single morning in a central Sarawak market. I have here first is the Salaka zalaka. This is a common one. The locals eat the fruit. It is commercially produced in Indonesia but wild sourced in Malaysia. And I also have here two different versions of Salaka sarawakensis. This is very popular among the locals. The fruits are extremely sour, but the locals use it as asam alternative to the more popular asam galugo from a different plant family. And we have also this from the rattan family is Ceratolobus concala. Now it's Calamus concala. This is sour sweet. It is not very common, very rarely seen. And we also have here a beautiful fruit bunch here. This is Elaiodoxa converta. Looks very beautiful. And you notice that the scale reflects fish scale skin, like skin. It is characteristic for Calamus palms. And lastly, we have the roots of Eugaisona utilis. This is wild sago, native palm. The locals split the outer skin to get the inner pith to stir fry as veggie. It's very bitter. So it is very important for us to conserve the environment and natural resources. The forest provides numerous resources for indigenous people and a potentially high export revenue for high-quality products such as rattans. Our project, Phenology and Pollination of Calamus, a study process, will hopefully shed lights into extending the usage of non-timber forest products. Many of the species are sourced as non-timber forest products. However, their silviculture, flora, biology, and what comes to pollinate these palms are little understood beyond a few key rattan species. This in turn causes poor management of economically important groups and lack of conservation in the wild population. Understanding their pollination biology is the key factor towards understanding the conditions they require in the wild and potentially open the way in novel plantation crop and at least better manage the population in the wild. Our project involves several days of direct observation. Up to 72 hours were conducted to establish the start and the end of the flowering time, flora movement, flowering stages, odor secretion, stigma receptivity, and pollen extrusion were observed. The pollination experiments were done to determine if the flower self-set. Floral visitors were observed together with its interactions with the flowers. Volume and concentration of the nectar were measured. The Brix reading is assumed a sucrose equivalent. Flowering starts in the early morning and lasted till late noon for female and male plants. There seems to be more male plants than female plants to encourage more pollination. Nectar was observed from the base of the petals of the male flower and the staminode of the female flower. The observed species are most likely apomictic. The floral visitors observed were quite diverse and consisted of several different taxa. Hover wasps, 2 types, stingless bees, 2 types, ants, flora mites, fruit flies, and even birds, which is the spider hunter. The flora biology of Calamus is relatively unknown unknown given its size, about 400 plus species. The knowledge of flora biology is important to a better understanding of this large genus. It also stands that the genus is heavily used in furniture, in fashion, and as well as in medicine. This is an example of a traditional medicine from Calamus. It produces analgesic effects in wound healing, anti-inflammatory effects, and who knows what else to be discovered. The knowledge gained from this study process can be served as a reference for other members of the genus to better understand the breeding process and to help in conservation of wild populations. This study process can be applied on the species Calamus draco, commonly known as dragon's blood or rotan gernan. Due to the over-harvesting of the fruits for their high economical value in pharmaceuticals and cosmetics, the population has greatly reduced in the wild. With the knowledge we gain from this project on the phenology and breeding mechanism on the rattan species, perhaps species such as Calamus draco, which is commercially very important, can be properly cultivated and the wild population of Calamus draco can be restored. Thank you.